I want to welcome you all to the 16th Annual IPN Retreat. It's hard to believe we've been doing this for so long. And every year it gets bigger and more exciting, so I'm really excited about the program for this year's retreat. I want to start off by acknowledging the sponsors that have given us support to allow us to bring the food and to the space. So in particular, we have a lot of support from GPS that allows us to do this. And then many of the institutions at the university, the Douglas, the Neuro, TOSI, HBHL, the BRAIN initiative, the BRAIN program, but also support from corporate sponsors this year, Blick Photonics. And so they'll be around. They have a poster at the poster session, so go and visit them. It's a cool poster about two-photon light sheet microscopy and Thermo Fisher Scientific. So thank you to the sponsors for your support. As usual, I just want to make sure to start off by acknowledging the team in the IPN. I think you've all probably had a chance to interact with the IPN office, and certainly they're there to help you guys out. So I just want to recognize all the work that they've done in the past year, and just let you know that they're there to support you during your studies. The Associate Director, Patricia Silvera, is here in the audience and will be here for the poster session, the award session at noon. Austin Milnerwood is also here. He's the new director of the rotation program. Within the IPN office itself, Catherine Vanka is the Student Affairs Officer. We have two Student Affairs Coordinators, Melissa and Priscilla. And so if you have any questions or issues that arise, they're probably going to be your first contacts. Mirko Sablich, who does admissions, and so probably all of you have had a chance to interact with him. And of course, Adriana Scambatera, who is the events and program and projects administrator, and she is responsible for this event today, so we all owe her a lot of gratitude. And then finally, of course, Mengting Hu, who's been running the China programs in the past few years. She's transitioning over to GPS as the programs start to run down. Okay, so a few announcements about what's going on in IPN this year. A number of interesting and important new things to report. So first of all, we had a lot of meetings with students in the past year, a couple of years, about stipends, and so we've been trying to implement and listen to your recommendations. As of this year, the stipend for PhD students will be raised by $1,500 above the base rate. So master's students will receive a little bit less. PhD students will get a bump, so it's good to fast track to PhD. Next year, that amount will go up by even more, and so the goal is to create a more sustainable support for PhD students, especially who are in it for the long haul. There's a harmonized top-up policy. So one of the big issues that was raised last year was that it wasn't clear whether everyone was going to get a top-up, how much the top-up would be. This year, together with the Faculty of Medicine, we've put together a set of policies which are on the IPN website about top-ups. And so if you receive a fellowship, you can talk to your supervisor about getting that top-up, which is now expected. And then finally, because it's gotten quite complicated with all the different levels of support, I sent out earlier this year a top-up stipend calculator, which can help you, you plug in the information about yourself and any awards you have, and it kind of spits out what the expected minimum stipend and top-up should be. Take advantage of that to make sure that you're getting the appropriate amount, and if you're not, have a talk with your supervisor or with us. Okay, the other things that have rolled out in the past year, the Neuroblog, which is a kind of a fun opportunity to highlight the science and the scientists working in IPN. So every month or so, we ask a student to write up a little description, not just of a paper that they just had, but of their trajectory, their personal story about how they got to that study. And so if Adriana reaches out to you and asks you if you'd like to contribute, please say yes. It's actually a fun way to just highlight your work. You can find them on the IPN website. We'll also be launching an alumnus directory. So we've contacted recent graduates from the IPN program from the last 10 years or so. And so the idea is to create a directory which is It'll be behind a security wall, so not anybody can access it, but all IPN students will be able to find this list where past students are now working, email addresses to contact them. If you're thinking of working in a particular industry or area, you want to find out what graduates are doing now, that'll be a good place to kind of get a sense of what's going on. And often it's really impressive where our graduates have ended up, and so I think that can also be a kind of light at the end of the tunnel. And then, of course, there have been a number of initiatives spawned from the EDI committee, including this year we ran a summer internship for undergrads in collaboration with COHUR, an organization that brings students from underrepresented backgrounds to work over the summer in labs. So this is kind of part of our recruitment efforts to target less well-represented groups. And then there's been changes that have been requested for the LOU, so going forward we'll be implementing improvements over time. I'm happy to announce that we've had approval now for the hiring of a second associate director for the program, and so hopefully we'll be able to provide more support to you guys. If anybody has, so I've contacted a few people and haven't had anyone take the job yet, so if anyone is either a faculty member who's interested in this position, or if you're a student who thinks that there's one person that you'd love to see as an associate director or future director, let us know, let me know, and I'll definitely follow up. I think that's a good opportunity to really learn how things work at IPN. And then we've also decided to start implementing something called the Super Mentors program. So up to now, every student is assigned a mentor, but the mentors are often just a formal representative of the program and they don't necessarily have training in dealing with crises, difficult situations. And so our goal is to bring together a small group of people who receive proper training in how to deal with student situations, conflicts in the lab, career improvement going on into the future, any kind of questions that you might have as a student. And we'd like that committee to be made up of both faculty and students. So again, if you're a student who's interested in providing mentorship to other students, get in touch with us and we'll try to add you to that committee. Additional training workshops. So we've been trying to set up collaborations around the world. We just have had for the last few years a program with Chengdu, China. Last year we were lucky enough to send 3 of the IPN students to a workshop on electron microscopy at the National Institute of Physiological Sciences in Japan. And we'll continue that collaboration with NIPS. In the next couple of years there'll be one most likely on synapses. And so chances to travel. We're also thinking about opening opportunities with the NIH and Singapore, and we've been in contact with the NIH about their programs. So chances to work in an NIH lab as a graduate student here. And finally, I'm happy to announce that the Bernice Grafstein lecture this year, which will be held on the 23rd of October at the Gene Timmons Amphitheater at the MNI, will be Catherine Dulac. Catherine is an amazing speaker she'll be speaking on the neurobiology of sickness and social behavior. She's someone who studies olfaction, sexually specific or sex-specific behaviors, pheromones. It's a really interesting talk, and she's a great speaker, so I recommend everybody attend. Just to kind of, for today's event, if you don't have a copy of the program, there's a QR code, you can grab it. They're also posted throughout the room outside. So we'll start with the keynote lecture to be delivered by Dr. Boyden, followed by Symposium A, which will be neuroimaging in the developing brain. There'll be a lunch session, which will be catered by the super popular Falafel Saint-Jacques. What I ask people to do is during the day, like up until lunch and the end of lunch, you can go visit the posters from poster session 1. So it's kind of a mix of lunch and posters. Take advantage of that time. After the poster session 1 ends, I'll ask the students who are in poster session 2 to put up their posters. We'll start the awards celebration, which is a kind of celebration of all the awards that students in the IPN program have received and the announcement of the Sievers Award winner, followed by Symposium B, which is multimodal neural imaging, biomarkers of Alzheimer's disease. and Symposium C, Novel Mechanisms for the Development of Future Treatments for Neurological Disorders. And so during that afternoon session, you can also drop by the posters from session 2, and then you can go and talk to people at the posters during the cocktail party afterwards. So there'll be cocktails and posters, which makes for lively discussion. Okay, and so with that, I'm very happy to introduce today's keynote speaker, Dr. Ed Boyden. I think we need to make that bigger. Sorry. So Ed Boyden is the Y. Eva Tan Professor in Neurotechnology at MIT. He's an investigator at the McGovern Institute and at the Howard Hughes Medical Institute. He's co-director of the MIT Center for Neurobiological Engineering. And I have a few notes about his trajectory. So, I mean, Ed has had a really remarkable rise. He did his PhD at Stanford with Jennifer Raymond and Dick Chen. And during that time, he helped to pioneer optogenetics, this technique that many of us are using today. And he's sort of an indication of Ed's propensity for thinking outside the box. More recently, he's been doing work on expansion microscopy and all sorts of new ideas that he's going to talk about today. So it's really going to be exciting. I mean, this idea of being able to do super resolution imaging without having to actually exceed the resolution of light is a very clever solution to a difficult problem. And I think you'll hear a lot of that in Ed's talk. Number of honors that Ed has received. There's a huge list. I can't go through them all, but of course he's been elected to the National Academy of Sciences and the American Academy of Arts and Sciences. In 2013, he received the Brain Prize, which is probably the most prestigious prize a neuroscientist can receive. The 2015 Society for Neuroscience Young Investigator Award, 2016 Breakthrough Prize in Life Sciences, and Canada's own Gardner Prize, Gardner Award in 2018. So Ed is truly impressive, and I think you're all going to enjoy his talk today. So Ed, please come up.
All right. Yeah, thank you so much for that wonderful introduction and the kind invitation to come and speak. And yeah, really looking forward to interacting with y'all and getting to know people and seeing what synergies might arise. Sorry, I was trying to add a slide just the last minute, but there's no more time, so. Okay, and then just make sure the display extends. And, oh no, too many things happening. Let's see if it just figures it out by itself. And it does. Great. Is it displaying? I can't tell. All right, thanks. Set my time. Great. Yeah, so it's really great to be here. And yeah, we're looking forward to chatting throughout the day and learning about what synergies might arise. I direct a group at MIT that works on technologies for imaging and controlling the brain. And I think what motivated me, I trained as a physicist and engineer to enter neuroscience, was the idea that neuroscience needs tools that directly confront sort of the fundamental difficulties in neuroscience. You know, if we think about different fields of biology, often there are key technologies that made that field advance. So in my friends who work on cancer, I mean, being able to sequence tumors and find the mutations and so forth, has totally changed how people think about cancer biology. In the brain, though, there's a couple things that I think make the brain particularly challenging. And one is the extreme spatial scales that you have to deal with on a daily basis. So brain cells are gigantic objects, right? In the human brain, they could be many centimeters in spatial extent. So by far the largest cells of any kind. The ones that go down our spinal cord, of course, have a meter of dimension. And yet the circuitry of the brain is minuscule, right? The wiring of the brain is nanoscale in dimension. And of course, all the biomolecules, right? Genes, gene products, and so forth are all nanoscale. And so this tension between the nano and the macro, in some parts of biology, you can approximate or gloss over it, but in neuroscience, you kind of have to be able to span that if you really want to look at how, say, the fundamental building blocks of neural circuits yield something as complicated as an overall behavior, right, or a pathology. And another issue, of course, is time. So space is challenging, but time is also challenging. So, of course, in many parts of biology, maybe you can look at a specific time scale and maybe ignore some other time scales, but in the brain we have extremely fast time scales to deal with, biologically speaking, right? Millisecond time scale electrical pulses within brain cells, chemical exchanges between brain cells, and yet a lot of the things that are interesting in neuroscience, right? Whether it's understanding brain aging or the language learning that occurs in childhood, or plasticity, and the list goes on. These things take a long time, right? Days, months, or even years. So today I'll tell you a couple strategies that we've been building over the years where we really, from day one, tried to think about the spatial scales and the temporal scales. Because if we built a tool that only worked at one time scale or only worked at one spatial scale, maybe we would not be able to understand how entire neural circuits work. And we share all the tools as freely as possible, but as I'll talk about at the end, we're starting to do a little bit of our own science as well. And so it's intriguing to think about whether we can integrate these tool sets, and maybe for small brains like fish and worms, could we try to build computer simulations of them in the coming years? Okay. So the first half of the talk will be about space, and the second half will be about time. And so let's start with the first half. So again, there's a big tension in spatial scale, right? Neural circuits are gigantic, but the building blocks of neural circuits are minuscule. And so many pioneering inventors have developed amazing methods for nanoimaging, electron microscopy, cryo-electron microscopy, super resolution microscopy, and the list goes on. But all these techniques, I think we'd all agree, are difficult to apply to large 3D objects, right? Like brain circuits. And even if you are able to apply them to a scientific question, a lot of them do require specialized equipment, which can be quite expensive. And so we started wondering, how could we sort of democratize nanoimaging and ideally make it cross scales? And so in our group, we often think about how can we be the opposite of what people have done in the past. So we started thinking, for literally 300 years, the way that biologists image small things is through some kind of lensing effect, right? You magnify the information, the image of the object, whether it's light or electrons or whatever that are being emitted or scattered or otherwise modulated by the sample. And so we started wondering, well, what's the most different thing we could think of? And we thought, well, could you make the object itself bigger? And so we thought of many ways of doing this, some quite out of the box, like whether we could detonate the brain and little pieces would go in all directions and you could reconstruct it. Not a good idea. We didn't try it. But the method that came to fruition quite rapidly once we pinpointed a few key insights was the following. Can we chemically weave a dense spider web-like mesh of swellable polymer, basically the stuff in baby diapers, and to do it inside the brain, right, not just outside, and furthermore, not just outside cells, but inside the cells, so this won't work on a living thing, and ideally in between and around the biomolecules themselves, right? Ideally, we pull all the building blocks of life apart from each other. And so two then grad students, Fei Chen and Paul Tilburg, tackled this, and we published this discovery that we could physically magnify objects in early 2015. So in panel B is a fairly macroscopic piece of the mouse brain, right? Several millimeters on a side. And in panel C, same piece of mouse brain tissue, about a day and a half later, we've blown it up 100 times in volume, about 4.5 times in each direction. And the polymer starts out super dense, like in the upper left. The spacing between the polymer threads is just a couple nanometers. And then when you're done, as I'll show you in a second, we've swollen the polymer and it attains an extended conformation, like in the lower left. So here's a movie showing a piece of the mouse brain being expanded. We formed the polymer earlier, and now you see us adding water about there. It's a time-lapse movie, so we sped it up from half an hour to half a minute. But I hope you can see that this piece of brain tissue is growing. We're making it physically larger. Now, all techniques owe a debt, of course, to prior discoveries. One paper that we found very inspiring was from an MIT professor named Toyuchi Tanaka. In 1980, he published this amazing paper on the phase transition-like physics of ionic gels. These are hydrogels full of charged branch polymers. When you add water, the water is drawn in through osmotic force, as shown in this cartoon. And then these polymer threads will swell, right? They have to move apart. But because the polymer threads are highly charged, we use sodium polyacrylate, which is covered in negatively charged carboxyl groups. You get charge repulsion, and you get these enormous fast volumetric transitions. And so we were very intrigued by this paper because the nice phase transition like physics, maybe it's a kind of process that could be a well-behaved and maybe even expansion process potentially. Now you can't just dump this hydrogel or polymeric material on top of the brain, you got to get it inside. And so, strangely enough, around the same time, 1981, there was a paper by Christian Dreyer and Peter Hausen where they synthesized polyacrylamide hydrogels throughout preserved specimens. So polyacrylamide is sort of the uncharged cousin of polyacrylate. And they were able to take embryos and tadpoles, form these polyacrylamide hydrogels throughout them, and showed that this could facilitate staining and imaging. So it's fun to think that if someone had put two and two together, maybe this could have been developed nearly a half century ago. Okay, so the core concept then is to take a cell like the one on the left and then make it look like the thing on the right. So molecules got pulled apart, and now they're like a constellation of molecules in 3D space, hovering like stars, separated from each other. To be more precise, we would like to have two molecules that were touching. They should now be some minimum distance apart. That's how you could conclude that they were colocalized, let's say. And then if two molecules were some other distance apart, it would ideally be scaled up by a linear factor. How do we make that work? Well, let's get into the chemistry. But before we do that, I just want to sort of get the frequently asked questions out of the way. People often ask me, oh, are you disrupting the sample? Absolutely. It is completely disrupted. People sometimes ask, what about the interactions between molecules? They're all gone. We're pulling them apart from each other. So just getting those things out of the way. Are we messing up the function? Absolutely. The specimen is not alive anymore. This does not work on living things. Okay, just want to get that out of the way, because people often ask that at the end of the talk, and I'm like, I just want to get that out of the way up front. But the good news is the information is being preserved, right? We're preserving the 3D relative organization of things, and that's by design, right? We designed the technology to allow this to happen through the steps I'll tell you about in a second, and then of course we validated it, which I'll show you after this movie. Okay, so how do we design it to be even? Well, we had to invent several chemistries to make this work. The first is we had to invent little handles or anchors so that we could pull the molecules apart from each other. So in this cartoon, the brownish molecules are representing proteins, and in purple we have the handles or anchors that we attach to the proteins so we can apply force to them, right? And pull them apart from each other. Next, we have to make that dense spiderweb-like mesh of polymer, so we can do that through free radical polymerization, not unlike that 1981 paper I mentioned. So white spheres represent the sodium acrylate monomers here, and they self-assemble through free radical polymerization into chains, and those chains then, when they encounter a handle, form a bond. So the polymer could swell and make force, and the handle could convey the force. So that's two of the core ideas. But we had to figure out one more thing. I mean, the molecules are often quite happy where they are. We need to loosen them up, and so we can use detergent, heat, enzymes, all manner of different strategies to soften them up or even eliminate molecules that we no longer need. And then when we add water, the polymer will swell, like I told you before, but now, because of the softening and because of those polymeric meshes and because of the handles, we can pull them apart from each other, the molecules apart from each other. Importantly, you can see that the topology of the polymer is sort of cross-linked. You can see that in this cartoon. The lines aren't independent, right? They are connected to each other. And so we think this gives a sort of an averaging during the expansion process to make it even more precise than you might think it to be. And so, yeah, we have validated this as well. Being this is biology, we should prove our claims. The design was meant to be isotropic, but now we have also done a lot of comparisons where we do pre-expansion imaging with a classical nanoimaging method, like a super resolution microscope. And then we do post-expansion imaging with a regular old, conventional microscope, the kind that is pretty ubiquitous in biology. And so here in pink, we did SR-SIM super resolution of some cultured hex cells with tubulin proteins decorated with fluorescent antibodies, because, of course, in light microscopy, we're usually looking at fluorophores attached to the target or some other contrast agent. We're rarely looking at just the biomolecules. In green is post-expansion after it's gone 4.5-fold bigger in each direction. And so we align the two through distortion, and then we measure how much distortion was needed to align them. And the answer is it's not zero distortion, but it's pretty low, maybe a few percent over a typical microscope's field of view. And it turns out that for almost all biological questions, that's okay, right? It's very rare that somebody's trying to confirm that the distance between 2 points is exactly 315.4 microns. Almost all questions are about the relative organization of things. But as I'll show you later, we and many others are trying to make the expansion process precise, and there are hints that it can be precise down to maybe even submolecular precision. So that's distortion. We also should validate resolution. Resolution is formally defined as telling apart two things when they're close by. Can you resolve them apart from each other? And so we can look at things like microtubules and other things of known dimension and see if we can, resolve those distances. And so here, with a 4.5-fold expansion, looking at things like microtubules, we got a resolution of around 70 nanometers. Now, a typical good microscope, conventional microscope, might have a lateral resolution of 300 nanometers. 300 divided by 4.5 expansion is about 70. So, if we expand more, can we get better resolution? I'll show you later that's possible. But even with 4.5-fold expansion, there are many things we can do that are difficult and sometimes nearly impossible to do with earlier techniques. And so, of course, the first thing we wanted to do was multi-scale imaging. Here, I guess I can try to, I can't use the mouse pointer because I can't see where I'm pointing. On the left is zooming from top to bottom. Each white square is zoomed into in the panel below. And the color code is shown on the left. We're decorating key proteins with fluorescent antibodies. And so I hope what you can see as we go down the left-hand column is that it's getting fuzzy, right? This is a piece of the mouse brain. And if you zoom in too far, your conventional microscope will hit its diffraction limit, right? So light has a finite size or wavelength, and you can't see things a whole lot smaller than that. Now, the right-hand column is the same as the left, same specimen, same microscope, but after expansion. And I hope you can see that it's much more crisp, right? In fact, this purplish fuzz in the lower left, you can cleanly resolve into pink and blue subdomains in the lower right. And those are synaptic protein densities. So blue is presynaptic bassoon, pink is postsynaptic Homer 1A. And then you measure the distance between these protein centroids, you get exactly the same number that Catherine Dulac and Zhaowei Zhuang measured many years ago, shown in the upper right, with storm superresolution developed by Zhaowei. But now you can make this measurement on a regular old confocal microscope. Now, when you blow something up 100 times in water, it's mostly water. And so as a byproduct of this, you get amazing transparency. In fact, it solves transparency in a way that gives a bit of a bonus, because you're not just evening things out, but you're kind of also setting it to be the refractive index of approximately water, right? Because everything else got diluted by 100-fold. And so you can do exquisite, for example, light sheet imaging, where you shine light from one angle and take pictures at a 90-degree angle. And so working with Eric Betzig, another super resolution pioneer, we took advantage of this. We expanded and then used a certain light sheet developed by Eric's group. This is some work spearheaded by Rei Gao, Sho Asano, and Gokul Padayula, working across the two groups. In the upper right, we have mitochondrial and lysosomal proteins being decorated with fluorescent antibodies. In the bottom, we're looking at myelination. But importantly, we can do it in a multi-scale way. And so here you can see, here, let's kind of zoom in for a bit here. So this is sort of a similar color code. We have a yellow fluorescent protein highlighting some neurons, and then pink and blue are synaptic proteins. Now we're zoomed in, really zoomed in there, maybe even too much. And you can get into, see individual spines and pink and blue pairs, which are synaptic, pre and postsynaptic components. But now you can zoom out. And so now you can see the inherent multi-scale nature of expansion data sets. That's in the mouse cortex. Eric at the time was at Janelia Research Campus, so a lot of people there work on the fruit fly, Drosophila. So here's a fly-through of the fly brain. These are the dopaminergic neurons of the fruit fly. The color coding is done by hand. But we zoomed in from the outside of the whole brain, and now we're zooming out to the lateral edges. And I hope you can see that you can see individual axons and dendrites really clearly, but then you can zoom out and get the whole brain. And so many people are now using fluorescent proteins and expansion to look at neural architecture. For example, using combinations of fluorescent proteins, the so-called rainbow strategy, developed by Jeff Littman and others. Here's an example where we're expressing different fluorescent proteins with viruses in the mouse hippocampus. And the white square in the middle is zoomed in at the top middle. So again, it looks blurry, right? The wiring of the brain is nanoscale for the most part, especially axons. And so in the upper right, though, is the same field of view, same microscope as the upper middle, but now you can cleanly resolve individual axonal branches. That's all protein visualization. What about other kinds of biomolecule? So when Oswasi joined the group, and later Gaoji Yang and Yi Kui made a very inexpensive version of this, we worked on anchors that could bind to other biomolecules, such as nucleic acids, and also sugars and lipids, and the list goes on. But here's an example with RNA. So we take a piece of the mouse brain, we can anchor and expand both proteins and nucleic acids away from each other. So you can see YFP, protein in green, and the pink dots are individual messenger RNAs being visualized after expansion with in situ hybridization with fluorescent probes. So now you can image individual molecules of messenger RNA with nanoscale precision in thick, intact tissues, which I don't think that was easily possible before. That's all about fourfold expansion, but you can expand more. Many people have relaxed or changed or improved the polymer recipe to get tenfold expansion. We have a paper with Laura Kiesling, led by Shuwei Wang and Tei Shin, coming out hopefully soon on doing 20-fold expansion in a single step. But eventually you run out of steam, right? If you expand enough, the thing falls apart. So if you want to expand arbitrarily large, one idea is polymerize and expand, form a second polymer in the space opened up by the first round, and expand it again. And so you can do this over and over again. J.B. Chang, when he was a postdoc in the group, we've worked out the prototype of this, and then Deblina Sarkar, Jin-Yun Kang, and Oswasi, with great help from Margaret Schroeder, worked out a really fast and inexpensive way of doing it. So the first version of the iterative method was a 20-fold method, so 4 times 4 gives you about 20. So a 300 nanometer lens would now have resolution of 20 nanometers or so. And this cartoon illustrates another feature of this, which is becoming more and more appreciated. So again, in light microscopy, we're visualizing often labels attached to target molecules. And so in the top row of this cartoon, the red and green antibodies can decorate proteins on the outside of a complex, but they're too big to get inside. If you expand proteins away from each other, though, like in the bottom row of the cartoon, now the blue and yellow antibodies can make their way in. So we started wondering, lots of biological structures, like synapses, for example, are extremely densely crowded. Could we make invisible molecules into visualizable ones? So this is kind of a dense slide, but bear with me for a second. So panels B, C, and D here are showing a calcium channel, RIM12, presynaptic proteins, PSE95, and yellow is pre-expansion staining with an antibody. There's not much yellow. And then pink, same antibody, same specimen, is being reapplied after expansion. And what you can see is that at these synapses, and we know they're synapses because we have a reference stain with some stains that don't change after expansion, we can see, for example, the calcium channel subunit, the RIM12, and the PSE95 at these synapses much better after expansion than before. Not all proteins are crowded. In F, G, and H, you can see Homer1, Bassoon, and Shank3, and there the yellow and the pink are more similar. So talking with Tom Lampied's group at University of Maryland, we wondered, hey, could we start to look at the organization of some of these pre- and postsynaptic proteins and look at their mutually nanoscale registration? And having been a grad student in Dick Chen's group and Jennifer Raymond's group, I was always very intrigued by looking at calcium channels and how they are organized in intact tissues. But back then, I did do some antibody staining, and it was difficult to get it to work, and maybe now you know why. Here now we're looking at CAV2.1, shown in pink, and then postsynaptic PSE95, shown in yellow, and we could look at the co-registration of the nanoscale organization of these. And so we were able to look at and find that indeed there is a co-registration with nanoscale precision between these presynaptic proteins and the postsynaptic ones, which has been hypothesized to be useful for the fidelity of synaptic transmission or plasticity. Now, having seen that you could take invisible proteins and make them more visible, Pablo Valdez-Quevedo, a neurosurgeon working with us, and Nino Kyoko's group, we wondered, what about invisible cells? Can we make them visible? Now, in brain tumors, aggressive tumor cells have traditionally had hallmark stains like being designated as Vimentin GFAP double positive cells, so you can use antibodies against Vimentin, shown in cyan, and GFAP, shown in pink. These are actual human glioma specimens. And the Aggressive tumor cells have classically thought of as, been thought of as Vimentin GFAP double positive cells. So the first couple rows, A, B, and C, the top 3 rows in the upper left are pre-expansion, and the bottom 2 rows in the lower left, same specimen, same microscope, are after expansion. And importantly, that fourth row, Pablo expanded, stained, and then shrank it back down. So if you don't want extra resolution, if you just want better staining, decrowding and staining and then collapsing can be helpful. And then on the right-hand side in panel G, in the right-hand side of panel G specifically, Pablo convened some scientists who then counted just by eye the number of Vimentin GFAP double positives. And with post-expansion staining and no swelling, right? Swelled, stained, and shrank back down, they counted 6 times more of these putative aggressive tumor cells than with pre-expansion staining. So is this a new cell type? Is it the same cell type that we were undercounting? Honest answer is we don't know yet. But certainly there's a lot of work to be done if that's how far off we are in terms of counting these. That's all in the brain, and maybe for the purposes of today's talk, I want to focus on the brain, but just to be aware, expansion can be used in other parts of biology. So, Yongshin Zhao and Otavio Bicure, when they worked with our group and a whole bunch of pathology groups, Astrid Wines, Stu Schnitt, and others, we optimized and applied expansion so it would work on a wide variety of human tissues of different kinds. So on the left are normal, and on the right are cancer-containing specimens from human patients. You can see the organ types on the left: prostate, lung, breast, pancreas. The color code is shown in the upper right. And I hope you can see here is that the post-expansion images do show more detail than the pre-expansion ones. So let's see. Let's try to summarize now. This slide really isn't meant to be read, but expansion is spreading pretty fast. I try to track every paper doing expansion experiments, but it's getting to the point now where maybe that's not going to be possible for much longer. But I think it's going to hit 800 at some point soon. It's being applied to the microbiome. It's being applied to fruit flies. It's being applied to the worm C. elegans. You name it. People have even started to expand tissues like bone by decalcifying it. And it's very easy to do. You can visit our website, expansionmicroscopy.org. Pre-COVID, we had lots of people visit, but post-COVID, of course, travel, I think, has kind of changed in culture. So in summer 2020, during peak COVID lockdowns, we posted photographic step-by-step tutorials on how to do expansion, including really rudimentary steps like how do you hold forceps or how to use a paintbrush to lift up a sample. And so I would estimate at this point that probably a couple thousand groups are doing expansion. Now, expansion is interesting because not only is it easy to do, but it's also easy to modify. And so something interesting is starting to happen. People are tweaking it for their own purposes. So, for example, I think I alluded to the expansion of bone, but J.B. Chang, who I mentioned earlier, he now runs his own group at KAIST in Korea, and he's been able to expand the whole mouse body, bones and cartilage and all, by tweaking the softening step, right? There's 4 steps: anchor, polymerize, soften, and expand. You can tweak the steps to your heart's content, and pretty much anybody can do it. It's just fairly simple chemical steps. We had been expanding proteins away from each other, but Silvio Russoli had an interesting idea, and what they did was take a protein anchored to the polymer, but this time, rather than expanding individual proteins apart from each other, they took a single protein anchored to the polymer, chopped it into pieces, and then pulled the pieces away from each other. And when you do that, the shape of the protein can be visualized. So here are the GABA receptors in the upper left. The top row shows some ground truth cryo-EM. And the bottom, you can see the fluorophore decorated GABA receptor. And the lower right, the expanded GABA receptor really overlays well with the cryo-EM ground truth. And so this paper is coming out in Nature Biotech quite soon. There's some nanobodies with the red dots being fluorophores on them just to help with the visualization. And then in a study that our group did not participate in, but I was really struck by it, work by Hans Donzel's group at IST Austria, they did a non-specific protein stain in conjunction with expansion and found out that they could do automated connectomic tracing. So connectomes are interesting, right? Could you get a full map of a neural circuit? But they're hard to get. I mean, up till now, they've been done with electron microscopy. Here now, they were able to do expansion with a protein stain and use machine learning to trace the connectome. So now it's intriguing to think that anybody could just make a map of a whole brain circuit. And then our group also works on extending the toolbox as well. Expansion is is nice in its own right, but it's also getting a reputation of being a great processing step before other technologies. So people are expanding and then doing mass spec imaging. People are expanding and then doing spatial transcriptomics and so on and so forth. And so just one example here where we expanded and then did in situ sequencing, one of the spatial transcriptomic methods. There are lots of them now, but you could expand and then run sequencing chemistry, right? And maybe this is a more fun example to look at. This is work collaborating with Aviv Regev and George Church and others, led by Shahar Alon, Dan Goodwin, Anu Sinha, Azwasi, and Fei Chen. We expanded a piece of the mouse brain, brought in some hybridized probes bearing barcodes, and then sequenced the barcodes. So we could do that with dozens of probes at a time. And sequencing basically just means you're copying the barcode nucleic acid. And as you do that, you blink out a fluorescent code. Anyway, there's not enough time to go into all the details, but it's all published. But the point I want to make is that you could look at libraries of messenger RNAs, for example, with nanoscale precision in synaptic compartments like dendritic spines. Okay, so we're at the halfway point. Let's summarize. We've discovered we can physically magnify objects. Now, ordinary microscopes can become nanoimaging machines. Also, decrowding molecules from each other makes the staining sometimes very vastly improved. It's a very easy process to do, and it's also a very easy process to modify, and lots of groups are tuning it for specialized purposes like protein structure solving or getting dense connectomics and everything in between. So yeah, very excited expansion, and a lot of my group still focused on trying to extend it and to make it even more powerful. But I think it must be all too obvious, and I tried to make it that way, it's not going to work on a living thing. And so in the second part of the talk, I want to focus on the imaging and perturbation of living things. Maybe we'll spend a couple minutes on optogenetics because it's kind of a very mature field. And just maybe I'll emphasize some new points about it that I think kind of maybe point in interesting directions for the future. But yeah, so in optogenetics, of course, we are controlling brain cell electrical activity with light. This started when Carl Beiseroth and I were both students at Stanford, and we just started brainstorming, how would you stimulate brain activity, right? If you could drive brain activity, you could test for causality, right? You could see if you could trigger a brain state or a behavior or a pathological state. And at the time, Carl was doing transcranial magnetic stimulation with his patients, and I'd trained as a physicist and been very intrigued by how could we think about tools for the brain? And so we started just by trying to think of every possible idea of how to do it. There's only so many kinds of energy you can deliver to the brain, right? There's sound, there's light. Light has a few flavors, right? Radio frequency, X-rays, stuff in between. And you can write down on a single sheet of paper pretty much every way of doing it, right? And so we thought light would be, of course, nice if you could get away with it because it's fast as anything ever gets and you can aim it really precisely. Just as electrical probes are brought into the brain, you could bring optical probes into the brain. And then I became really fascinated by this class of molecule known as microbial rhodopsins. So these are 7 transmembrane proteins, like the one in the lower left. The first to be found was a light-driven proton pump that in single-celled microbes would pump protons in response to light across the membrane that then powers the production of ATP. And so it's sort of a primitive form of photosynthesis used by archaea and other single-celled microbes. Later, people found light-driven chloride pumps, also from single-celled microbes. And then later, people also found light-driven ion channels. The channels, of course, are not being used to store energy. Channels will dissipate energy. But they're used by single-celled algae, for example, to help them navigate around in bodies of water. And so just to flash forward to the current day, what we and our colleagues and collaborators have found is that members of all of these classes can be found that serendipitously are safe enough and fast enough and strong enough that you could use them in delicate brain cells. And really, it was a lot of serendipity. I mean, we now can put the light-driven proton pumps or chloride pumps into neurons, shine light of the right color, and they'll hyperpolarize brain cells. Work done by Brian Chow, Xue Han, and Amy Chuang. And on the right-hand side, Carl and I put a light-driven non-specific cation channel, CHR2, into brain cells and published that together in 2005. And later, Nathan Klepetke and others have been trying to perfect the toolbox, meaning can you get the molecules as fast as possible, red shifting to reduce the need for delivering high power that could heat the brain, and so on and so forth. So I think that's all I want to say about optogenetics, just because it's so mature, and a lot of the molecules that are still in use today were published before 2012 even. But it's fun to think about one recent story that's starting to rise in some prominence. You know, up till recently, optogenetics was primarily a tool in animal models like mice. But a couple summers ago, a European team led by Boton Rosca and José Alain Sahel, and they co-founded a company, GenSight, to lead these clinical trials. It's hard to do a clinical trial in academia. But they took crimson, this molecule that Nathan published in 2014 in our group, and they showed that patients with retinitis pigmentosa, where they lost the photoreceptor cells and therefore are blind, they can get a pretty good, not perfect, but a decent partial restoration of functional vision. So this gentleman here was able to recognize some household objects. He could see lines on a crosswalk or recognize doors along a hallway. Couldn't recognize faces very well, had trouble reading text, so it's not perfect vision, but you know, to my knowledge anyway, it might be one of the best restorations or the best restoration of vision for this condition to date. And Boton and others have lots of ideas on how to improve it even further. And so maybe I just want to talk about some newer things now, but just to emphasize how lucky optogenetics was. You know, the molecules could have been toxic. They could have been, you know, who knows, misfolding in neurons, or, you know, with the wrong performance or whatever, but they weren't. And maybe the most, and maybe it's an underappreciated stroke of luck, is that, you know, there's an all-trans-retinal, a vitamin A variant that is the actual light absorber. And for whatever reason, the human body and the mouse body and the mammalian brain and so forth have that, which is sort of interesting. It's not true for all brains, right? You know, the worm C. elegans, for example, or if you look at, you know, you have to feed the all-trans-retinal to the worm. It's not found naturally in it. So, yeah, if that wasn't a happy coincidence, maybe it would have been a lot less useful. And so I just want to spend the last bit of the talk talking about some newer things we're doing, though, on sort of the opposite direction, the readout of cellular signaling in the brain. Now, we got super lucky with optogenetics in the sense that these molecules out of the box, with basically zero modification, were able to function with no chemical supplementation in the living mammalian brain and so forth. And so for reading out signals from the brain, we haven't been quite as lucky. But human ingenuity has been filling in that gap. And so hundreds of fluorescent biosensors have been engineered by wonderful bioengineers, where you take a fluorescent protein like the GFP from jellyfish or red fluorescent proteins from coral, and you fuse it to a sensor domain, like a calcium binding protein, and then you can make a fluorescent calcium indicator. You just put the gene in for that protein that fuses the calcium binding protein and the GFP, and now the gene product, the protein, will light up when a neuron is active, for example. And so this is a screenshot from Jin Zhang's wonderful database from her group at UC San Diego. And people have made all sorts of fluorescent biosensors that are genetically encoded for protein kinases, for second messengers like cyclic AMP, and the list goes on and on. And we've been doing some ourselves as well. Christina Torres-Gaben, who I mentioned the lower left here, worked on a potassium indicator when she was in our group. Kiro Pijakovic and Erica Jung, when they were in our group, worked on fluorescent voltage indicators. It's been a really fun adventure to be part of that community. As time went on, though, we started wondering, cells have a lot of stuff going on, right? I think we've all seen cartoons that look like this, right? You know, this is a schematic of a cell. On the left, we have some cellular inputs. They could be nutrients or hormones or modulators or transmitters that are landing on the cell membrane, shown in orange. And then inside the cell, in the middle of the slide, you have all these kinases and transcription factors and motor proteins and other things that are in a complex network, right? I mean, we talked about connectomes and brain science at the macroscale level, but there's almost like a molecular connectome, right, if you will, inside the cell. And then in the nucleus, shown in blue, then maybe you have some kind of output, right? The cell might turn on or off a gene, or maybe it decides to, you know, live or die or whatever, right? And so each of these arrows that connects one molecule to another might have been a whole PhD, right? When I was a grad student, I worked on some of these pathways, actually. You know, but knowing the whole network for a given cell is tricky, right? These cartoons are, in some ways, a little bit fictional, right? For any given cell, maybe only a fraction of this network exists in it, right? how would you validate the network in a given cell? And maybe in a disease state, that network would change, right? So we started wondering, could we just image a whole network? Could you look at the inputs to a cell and the intracellular computations and the outputs all at the same time? And so we thought about this for many years, trying to think about what we could do about it. And we put forth kind of the same kind of constraints that we always put forth for our group, which is, okay, this needs to be easy to use and simple. We never want to make a technology that's too hard to use, right? And so this was the constraint we put forth. You should use ordinary microscopes to do it. And just as optogenetics, we got lucky. It's fully genetically encoded. You don't have to add the vitamin A variant even. Ideally, we would also be fully genetically encoded. And so it's interesting to think about. One theme that I recently realized our group might have is we take a classical engineering idea and we implement it through chemistry. So nanomechanical engineering, that's expansion microscopy. Tiny solar panels, that's optogenetics. And so we basically took inspiration from the internet and from cell phones and decided to try to implement how the internet works and how cell phones work with chemistry. And so one idea is called spatially multiplex imaging, and the other is called temporally multiplex imaging. Now, spatial multiplexing, let's see. In this building, there's probably like, I don't know, 50 or 100 or more Wi-Fi antennas, right? And if you walk around from room to room, your cell phone or your laptop is going to go from one Wi-Fi antenna to the next. And that way, this building can support way more cell phones and laptops than if there's only one antenna, right, that we all have to fight over, right? That's how cell phones work as well, right? In fact, the name cell phone comes from the idea that as you go around a city, you go from one cell base station to the next, right? And so we started wondering, what if we could take a biological cell, not a cell phone, and what if different places in a biological cell could tell you different stories? I mean, a molecule is much smaller than a cell, so a cell could potentially report on many different things if different places could broadcast different information, right? So I'll tell you how we do that in a second. And the second idea is temporal multiplexing. So all of our phones use fairly similar these days frequency bands to communicate, but they each have a different temporal code, which in some ways makes them orthogonal to each other, right? So they can be sorted out. Old-fashioned AM radios, right? They have different frequencies, and different frequencies then can also coexist in the airspace over a city, and the AM radio can then extract just one frequency and ignore the rest, right? So temporal multiplexing is also a core concept of all modern telecommunications. So in the last bit of this talk, I want to tell you how we make these things work through chemistry. Okay, so let's start with spatial multiplexing. It's easier to explain. So when Shannon Johnson and Chengyang Lingu are in my group, we tried to figure out how to make this work. So imagine, as shown in this cartoon, points labeled 1 have one kind of fluorescent indicator, maybe a calcium indicator. The points labeled 2 would have a different indicator, like a protein kinase A indicator. So while the cell is alive, little clusters of indicator will be randomly and stably sprinkled throughout the cell. And while the cell is alive, they'll blink out little movies of what's happening at that point. And so you want the points to be dense enough to sample the biology, but far enough apart that you can separate them, right, in a microscope. And then when you're done, you can preserve the cell and use staining techniques, kind of like the stuff that I talked about in the first half of the talk. And you can stain, image, wash out the stain. You can do this over many cycles, right? Because once a cell's preserved, you can kind of do whatever you want to it. That's the concept. So we tried many ways of doing this. It's actually really hard to make this work. We had many false starts. But in the end, the way that worked the best was the following. Take different fluorescent reporters and fuse them to different self-assembling peptides. These are proteins that act like the coat proteins of viruses. They self-assemble into clusters, right? Except these are human designed, so they're bioorthogonal. They're not going to hurt the cell. Then, different reporters will self-assemble, the design goes, into stable but random points in the cell. Also, each fluorescent reporter has a different epitope, basically something we can stain for, so that later, when the experiment's done, we can figure out which sensor was where. And so to cut to the chase, it works. Here we took 3 different fluorescent sensors from other groups, the much-loved calcium indicator, GCAMP6, a cyclic AMP indicator, CAMP-R, the pKa indicator, X-ray ACAR. They're all green fluorescent protein-based, by the way. So if you express them all in the same cultured neuron, as you see here, you get the green image while the cell's alive, and those dots will be blinking at you. The false color images are what you would get after you stain, after you're done with the experiment and preserve the cell. So while the cell's alive, you get traces, and then later you can go back and figure out what each trace meant when you do the staining. So when I was a grad student, I was working on learning and memory with Jennifer and Dick, and there's a whole class of sort of intellectual structure of question, which I got obsessed with at the time, and wanted to really build a tool to help confront it, which is how does the brain convert a short-term stimulus or event into a long-term change, right? You know, a brain injury might cause long-term inflammatory and other changes. Maybe, you know, learning, of course, a brief exposure to a stimulus, and the brain will remember that, you know, years later, right? We all have memories from childhood, you know, that are effectively permanent from a human standpoint. And the list goes on and on, right? There's lots of questions in neuroscience that have this flavor. So I really wanted to see if we could build a tool that would let us follow things over time, right? Getting back to the theme of temporal scale. And so this is a cultured neuron, so it's not going to undergo learning, but we can do sort of pharmacological models, right? You know, long-term potentiation, long-term depression that have been studied in vitro for many decades. And in this case, we thought, let's do a quick check. We'll give forskolin, which drives cyclic AMP production, and we'll image calcium, cyclic AMP, and PKA at the same time. Now remember the goal, we want to map out the network, right? We'd love to see how they all relate to each other. If you just image calcium in one set of cells and PKA in a second set of cells, you will not be able to see how they relate to each other, right? Or at least it'll be much harder to figure that out. And so we were able to find some potential linkages between these. So in panel H, some neurons had delayed calcium responses to forskolin, and in panel I, some neurons had faster responses to forskolin in terms of calcium. And so PKA activity stays on after you take the forskolin out. You could think of it as sort of one of the classical outputs of this molecular circuit, so to speak. And so we wondered, can we predict anything about PKA from the calcium? And so focus in panel L, the fast calcium neurons had strong PKA activity, and the slow calcium neurons had weaker PKA activity. And so, and we found similar results when we repeated this in acute brain slices of mouse hippocampus. So we were excited that we could see the relationship, right? How the speed of one signal relates to the amplitude of another. And we could add a fourth indicator. Here's a PKC indicator. We could throw in a fifth indicator. Here's an ERK kinase indicator. And so this goes on and on. You know, right now I think we're basically limited just by the number of genes you can put into a neuron, which is something we're starting to try to figure out. You know, could you put 20 genes into a neuron? But that's going to take some time to figure out, I think. So we're very excited about this, right? We could take lots of these off-the-shelf indicators, park them at different stable places in the cell, and we did lots of controls, by the way. This is all published, so I didn't make slides on them, but we looked at cell physiology, we looked at cell health, we looked at sensor quality, and none of that was different from when the conventional sensor was used. And so I want to end just by talking about temporally multiplex imaging in the last several minutes. This is a bit harder to explain, and I'm still trying to figure out the best way of explaining it, but I'll try an analogy, and then I'll try a direct mathematical explanation. So the analogy that I would like to make, us being neuroscientists, would be to how the ear works. So sound enters our ear, and then our cochlea, the way it works, of course, is you put different frequencies, at different places in the cochlea, right? So the cochlea is a spiral, and high frequencies will end up driving neurons at a different place than low frequencies, right? And that's the basis for sort of the earliest spectral components of hearing. And so if you listen to a piece of music or you hear speech or whatnot, your brain is sorting out those different frequencies. They're linearly independent, right? So what if you could give each molecule in a cell a different musical instrument, right? You can imagine taking a brief movie of the cell. We're not going to use actual music, right? We're going to use light, of course. But imagine you have fluctuations that act, in some kind of periodic fashion like musical instruments. You could take a movie of the cell, do the same transform that the cochlea does, a Fourier transform, and you could then unravel all the different components. Now, how do you do that? Well, it turns out there are flickering fluorophores, reversibly switchable fluorescent proteins. And so in work that Yang Chen did when he was a postdoc in our group, we would take different switchable fluorescent proteins. Here are 6 different greenish ones shown at the bottom. Now, what happens? Let's just focus on this for a second. So you shine blue light on one of these green RSFPs, reversibly switchable fluorescent proteins, and they'll glow green, but they're also going to go dark, right? Note well, this is not fluorescence lifetime. This is not photobleaching. This is reversible photoswitching, a different kind of thing. And you can use blue light, or purple light, excuse me, to bring it back to the bright state. If you just shine constant blue light, then they all go dark with an exponential decay. And in music, maybe you think of sinusoids as being sorted out by our cochlea. They're linearly independent. Exponentials are also linearly independent, right? Because exponentials and sinusoids are very similar mathematically. And so, what if you had a cell with a bunch of these fluorophores in it? You could take a brief movie and then basically invert the movie and recover all the different components. The same way that our ear can extract all the sounds from a piece of music. Okay, that's an attempt to make an analogy. Now let's just talk to the math, I guess. So let's have a couple cells here, and each cell has a bunch of different reversibly switchable fluorescent proteins. Maybe they're fused to different proteins you want to watch. Maybe they're just being expressed under different genetic promoters. So the spatial multiplexing works well when you have reporters ready to go, right? But what if you want to just look at where a protein is? Well, this is where the temporal multiplexing could be helpful. So, excuse me. So while the cell is alive, each point in the cell will have some combination of these RSFPs. At the top right, each RSFP will have a different characteristic exponential decay. And then going down the column of the right-hand side, if you take a brief movie of the cell, each point in the cell will be a linear combination of those exponential decays. And the coefficient of each in the linear combination is the amount of fluorophore there, and therefore the amount of protein or the amount of gene expression. You can use a standard method called linear unmixing, and you can then separate the sum into the components. Modulo the noise, right? Any weighted combination of exponentials can be decomposed into those exponentials in exactly one way, right? Okay, I've tried to explain it 2 different ways, but maybe let's get to some data. So in panel C, we took 6 different green proteins with different time constants of switching, fused them to different proteins of interest, an ER protein, a tubulin protein, a lysosomal protein, and so forth. Took a brief movie, did the linear unmixing, and now you can get all 6 parts of the cell with one color. That's anatomical, but you can also look at dynamics. So because you have to take a brief movie, you can't do ultra-fast imaging, right? We can't do calcium imaging yet with this. But, we could look at the cell cycle, for example. And here we took 4 different proteins that turn on at different parts of the cell cycle, fuse each to a different RSFP with a different time constant. We could take brief movies every now and then, invert them each. So each row here is a different movie taken at a different time. And then each movie, when inverted, gives you a single set of images. And you could look at cell division. And it's a lot of fun. Every cell has its own little personality, and we can already start to map out parts of the cell cycle in greater detail than before. So I'm not an expert in the cell cycle, but at the time, it looked, at least to my untrained eye, that CDK2 and CDK4/6, these are cyclin-dependent kinases involved with the cell cycle. They'd been studied in some parts of the cell cycle, like M phase and G1 phase, but it looked like in S phase and G2 phase, we didn't know what they did. But now you could look at, in this case, 6 different things at once using this strategy. Each of these panels in the lower left is an individual cell, so you can really start to look at how every cell is so different, right? It's really fascinating just to think about what the cell's going through as it tries to decide when and how to divide. Anyway, in panel C, we found that CDK2 activity went up during S phase, which wasn't known before. And then CDK4/6, as shown in the lower right, did not go up during S phase. So again, I'm not an expert in the biology of this, but it was fun that we could make such discoveries even just by fairly simple experiments. And so I think I'm going to end the technology parts of this talk now and headed to the wrap-up part. We've discovered that there are ways to use basically telecommunication theory implemented through chemistry to image many things at the same time while something is still alive. Spatial methods, temporal methods, again, shamelessly borrowed from how the internet works and how cell phones work. And they're really easy to use. They can work on ordinary microscopes with no fancy hardware, and they're all fully genetically encoded. So again, ease of use is always a big deal for us. We always want to make something fully democratized, ideally from day one. And all of our tools are shared freely. The optogenetic tools and the expansion tools are being used by thousands of scientists each. The live multiplexing stuff is newer, so that stuff is still making its way into the world, but it feels really exciting right now. It feels a bit like optogenetics in 2008 or expansion microscopy in 2018. So if you want to try this out, all the constructs are freely available, or you can always drop me a line. So recently we also started thinking, we've been sharing these tools freely for, I guess, wow, almost 20 years now. Seems like a long time. But where are we all headed, right? And so I started out as a physicist and computer scientist, and I really was thinking a lot about how do we understand brain circuits, and maybe having trained as a physicist, we often think of solving something very small, very completely, right? In physics you study the hydrogen atom, right? Or something very, very simple, but you really understand it very, very well. And so we started thinking, what if, from bottom to top, we could image lots of signals throughout an entire brain while it's alive, and then going up, we could then use optogenetics to perturb key nodes in the network, and then finally, when you're done, preserve the brain and do expansion. And then you could do the spatial transcriptomics and map all the cell types, and the connectomics and map all the connections and so forth. Could you make a computational model of an entire brain circuit? And so it's early days, but we've quietly been working on sort of getting things going over the past 10 years or so. So when Limor Freifeld was in our group, we adapted the expansion method to the larval zebrafish brain and showed that we could expand the whole larval zebrafish brain. When Jay Yu was in our group, he optimized and showed that we could expand the worm C. elegans. So those are the two species we focused on, worms and fish. And then things like voltage imaging, we've also been working to adapt for these organisms. So in 2018, Erica and Kirill, who I mentioned earlier, developed a voltage imaging reagent, and then we adapted it to worm imaging and fish imaging. And most recently, Ziguan Wang in our group built what might be one of the world's fastest light sheet microscopes. We could scan the whole larval zebrafish brain, expressing a voltage indicator pan-neuronally, and image the whole fish brain at 7,000 frames a second, so 200 volumes per second. And so you can see here a lot of traces on the right-hand side that look like electrical traces, but they're not. They're all voltage imaging traces. So I really think voltage imaging is ready for prime time. It's really, really exciting right now. And then, so our goal is to combine these data sets using deep learning and physiological modeling, inspired by examples like Eve Marder and others who have been able to take complete data sets for very small neural circuits and get extremely accurate models of how they work. Can we apply them now to entire small brains? But we've also been thinking hard about what's the path to scaling these tools up to whole mammalian brains. So some groups have several groups actually, have expanded entire mouse brains. And even the Allen Institute has showed they could expand the whole mouse brain, and then with a custom light sheet microscope, scan the whole darn thing without even sectioning it. So preserved tissue physics can be hacked and tweaked, right? Because you can do almost anything you want to a preserved tissue. But we've been thinking a lot about, well, what about the optogenetics? What about the imaging of physiology? That's much harder to scale to an entire large brain, like a mammalian brain. Even scanning the whole zebrafish brain is challenging, right? This preprint is heroic, but it just shows how hard it is too. And so it worked that we just posted on bioRxiv a couple weeks ago. We put forth what might be the first attempt to make the living mammalian brain more transparent. And then another group posted a preprint a few days later. So it's great to see that multiple people can get this kind of thing to work. But Giovanni Tagli-Francesi and Ishan Gupta are the co-first authors of this. But by using common biocompatible molecules, in fact, ones that are even used in human subjects, like high molecular weight dextrans, the idea is let's shift the refractive index of the saline, the solution around brain cells, upward a bit so that the water-lipid interface gets smoothed out. And so both our group's preprint and then Takeshi Imai had a similar thing using a different high molecular weight biomolecule. Both got sort of similar results in terms of improved 1-photon and 2-photon imaging of calcium dynamics and also voltage imaging in living brain circuits, including the living mouse. And so it's early days, but the fact that multiple groups are getting interesting results with different molecules suggests that we're something, a whole new area might be opening up. So I think I'll end there. I think I've acknowledged all the people who've led these projects along the way. Please visit our website, synthneuro.org, to learn more about the tools. And then I'm the PI of the group, but also the chief tech support person. So just email me at boyden@mit.edu if you have questions. This slide also lists lots of people I didn't have time to acknowledge along the way for these and many other projects. including alumni and other group members at the top over the last 20 years or so. And then the middle of the slide, an even longer list of just the PIs of groups we've worked with over that period. So I'd be happy to take questions now.
Thank you, that was great. Are the microphones working?
Excuse me.
Well, if you have questions, go to the microphone. Or not. Actually, why don't you just raise your hand? We have time for a few questions. Yes, go ahead. Speak loudly.
No.
No.
Hi, Doctor. Thank you for the excellent talk. Just curious about the expansion. So can we tell the damage after the expansion? Any marker to tell us what damage happened after the expansion?
What kind of damage?
Just curious because I am late for this meeting, so my partner missed this part. So will we do expansion? Will we break the circuit of the neurons?
Yeah, so I made this slide because I always get this question. Yeah, so the specimen is completely damaged. It's just complete damage. Yeah, but the information's still there. So yeah, it's no longer alive. All the molecules have been pulled apart, and the molecules are no longer interacting, and there is zero function left. But the nanoscale organization has been preserved, and so that's why it's popular, because you get the information that you want, right? Which is the organization of the molecules.
I see. Thank you.
Yeah. Great question.
Right here. Testing. Thank you so much for the talk. Like, it's one thing knowing expansion microscopy and another thing hearing you talk about it. I have more maybe a naive questions. I'm really interested in calcium channels, and I saw that you showed an image of it. I usually use we would use super resolution microscopy as a ground truth in this case. But you're showing that we're able to get more staining using expansion microscopy. Now, in that case, if I'm going to do an experiment staining for calcium channels, which one do I use as ground truth? Can I see that expansion microscopy now is showing me an artifact? Or how do I tell that? And what's your thought on this question?
Yeah. So we did a lot of careful controls to try to see if indeed the calcium channels are artifactual. So first of all, they show up at synapses, right? If it was an artifact, it'd be very weird if every artifact somehow magically knew where synapses were and then went to the tiny fraction of the brain that is occupied by calcium channels at synapses. So we did a lot of controls, as shown on this slide, to look at that. Sometimes we'd be lucky, and proteins are finite size. So sometimes you would have an antibody that binds one part of the protein, and it would work fine pre-expansion, but a second antibody would not work. And then after you expand, suddenly the second antibody works. So that's what happened with PSC95, actually. We had two antibodies, one that worked and one didn't. After we expanded, both worked. But here's the thing, right? I mean, antibodies always need to be controlled for, right? How do you know that an antibody doesn't bind other things? There's really no way to know that without doing good controls. And so I think the maybe more cautious answer is that all antibody studies should do controls, ideally. I guess if we were trying to be super rigorous, you could imagine using blocking peptides or knockouts, but a lot of people don't do that. There could be proxies for that too, though, like making sure that one is using an antibody that's been thoroughly tested in the past, ideally by methods that have some kind of causal intervention. But yeah, it's interesting that a lot of the antibodies get better with expansion. The other thing too is that expansion has also gotten to the point now where so many people are doing it, right? So that's why I showed this slide. That when people email me about expansion, I can often be like, oh yeah, in that field there's already 10 papers that have done this kind of thing. You can go talk to them too, right? And so the good news is that now 9 years into this adventure, you're not alone, right?
Definitely.
And post-expansion imaging is one of the fastest growing parts of expansion, in part because of this idea that invisible things could be detected.
Exactly.
So lots of people are now using post-expansion imaging to look at things. unveil all sorts of interesting features. So,
Definitely.
but yeah, I do think good control should always be done ideally for any experiment, not just for expansion experiments.
Yeah, definitely, because there are some calcium channels that are appearing at places that we never thought they would appear. But all your advice are really sensible. Thank you so much.
Well, for calcium channels, I think you'd also look at like GFP fusions. I mean, there's lots of, because even a normal, like you use the word ground truth, which I love to use as well. But there's also, I think, it was a Nature Methods article about 10 years ago about how even just fixation, right, and permeabilization alter proteins, right? And are you losing a post-translational modification, or, so live cell imaging or preserved cell imaging where you did a GFP fusion, and there are also knock-ins. I mean, there's a whole field of this, right? So I don't want to belabor the point, but I think that there's sort of like standard ways of thinking about proteins that are definitely applicable here.
Definitely an exciting era. Thank you.
Thanks. Yeah, that was a really fascinating talk and really incredible images. I'm wondering, also about the idea of having epitopes more accessible after you expand the tissue. Does that also mean it's a lot easier to wash out stains? Like, do you think this is something that's going to help for sequential immunofluorescence when you're, staining and washing out and things? Is that being looked at?
Yeah, so we have a paper that just got accepted, led by Margaret Schroeder and Jin-Yun Kang on multiplexed staining after 20x expansions. So now they're doing 20 quote-unquote color imaging with 20 nanometer resolution at 20-fold expansion.
Wow.
And it's great, yeah. So we could expand like amyloid plaques and look inside to try to figure out where glutamate receptors might be hiding inside of them. Yeah, it's laborious because you have to stain, image, and then wash, which can be painstaking. Maybe somebody will automate it someday, but it does work. And is there a fundamental limit on how many one could look at? I don't know. Maybe not, right? Because if you can, and also there's a whole field now where people are barcoding antibodies. So take an antibody, put a piece of DNA on it. Imagine you put in 100 antibodies with 100 pieces of DNA, sequence the DNA right there inside the specimen, right?
Yeah.
And one of our collaborators just posted a bioRxiv preprint yesterday on expansion genome sequencing, where they're expanding the nucleus and then sequencing the genome right there inside the cell, right? And so, which our group helped them with. So, so if you combine these ideas, then it's very intriguing to think of the idea you could just expand an object, sequence everything, and read out the genome, the transcriptome, and a good chunk of the proteome at the same time.
Yeah.
Nobody's done that combination yet, but I think this most recent paper suggests it would be possible.
Great. Yeah, looking forward to seeing it. Thanks.
Thanks. Just to stay on schedule, I think we're going to have to end the questions. However, it is planning to stay for the entire
I'll be around
meeting, so
all day, so.
Please, feel free to approach them with questions.
Great.
Thank you so much. So now we'll take roughly 10-minute, physiology caffeination break and then come back for the next session at 11 o'clock sharp, please.